cool. Proteins in the early 1900s. In the early 1900s, they actually thought proteins was the molecule that carried the genetic information. They knew more about proteins. They knew about amino acids. They were starting to discover and get into proteins. They looked at proteins and they said, oh, look, there's 20 different amino acids. Like we have 26 letters of our alphabet, here are 26 molecules that can function as kind of a letter or signal of a certain thing, and you can write information in these molecules. And so they said, well, that's pretty good. You can write a lot of words with 26 letters, right? Get over to the library and get a big fat dictionary. However many hundred thousands of different words are in there. They already knew about DNA. They knew about nucleic acids. They knew DNA was made up of 4 different nucleic acids, A, T, G, and C. But they thought, how can you can't transmit very much information with only 4 digits in your alphabet. Follow me? This was the thinking. That's not going to be able to transmit the amount of information that wherever that genetic information is housed. Didn't seem like you could do it with a molecule that has only 4 different things on it. But DNA is so long, and you can have ATC, ATG, GTC. You can have the order of those can be so mixed up when you got literally millions of them along the DNA molecule that you can code things. And then, of course, we come along with our computers. How many digits does your computer use? 2. 2 digits. Your computer is dealing with circuits that are either open or closed. So that pathway there is open or it's closed. But there are many different pathways. And you put that open and closed sequence in order, and you can do what you can do with computer programming with only 2 signals, an open circuit or a closed circuit. So you can do quite a bit with 4 digits in DNA. Well, okay, now I'm saying all that to show you how protein was so attractive as a molecule that could really transmit a lot of information. I don't want to downplay proteins. They are extremely essential molecules. And in fact, 10 or 12 of them are needed to maintain the DNA molecule. So the DNA molecule requires protein to maintain it, to unzip it, to repair it, to synthesize it, to replicate it. There are protein molecules involved in all those processes. So proteins are really, really powerful and really essential molecules in the body. Now, you have about over 3 trillion. Every time I go look at how many cells are in the human body, it seems like it's gone up a couple trillion. But I've seen numbers like 3 to 10 trillion cells in your body. All right? Trillion. Every one of those cells can make about 10,000 or more different protein molecules. They all have the ability to make thousands of different protein molecules. And what do these molecules do? Regulation, enzymes, biological catalysts. And when I say make, the protein has to be made inside the cell. We're not making proteins outside the cell. There may be some protein fragments floating around in the interstitial fluid between cells, but essentially making and using and the function of proteins is going on inside the cells of your body. So if a particular cell, the cell of your eye or your stomach, needs a certain protein, it has to make it. That cell has to be able to make it. So biological catalysts, that'll be chapter 19. They are proteins. Transporting materials, we saw the cell membrane. There are proteins embedded in the cell membrane to enhance or to allow for things to transport across that cell membrane. Movement, there are certain nutrients. So proteins are doing just about anything your body does, it's utilizing proteins in that process somewhere. Okay, now they're structurally similar, and the building block of proteins are amino acids. Amino acids. And from your organic chemistry, we'll see this on the slides there too, but on your notes, at least draw this. We're going to have a molecule, the acid. I probably should have put here the amine first. So it's an amino acid. All right, so what do you see on an amino acid? You see an amine and a carboxylic acid on every amino acid. And then the other thing you're going to see is a hydrogen right there. There's 3 of the 4 things. And they keep picking on my initials. But then there's an R group right here. What is R? You define R, 20 different R's, you have 20 different amino acids. This is the general structure of all of the amino acids. And you're going to see that they're referred to as alpha amino acids. And way back in chapter 14 or so, I showed you a slide looked like this. Some of you might remember it. And I said there is a kind of another name nomenclature that would, particularly with carboxylic acids, that the carbon next to this is considered the alpha carbon, the beta, the gamma, trying to remember my Greek alphabet. So this carbon right next to this is your alpha carbon. This is why you will see this nomenclature sometimes. We're going to just call them amino acids, but they will also be called alpha amino acids. I told you that the amino acids and the saccharides have some pattern to them. They're a little bit more organized than lipids. I kept pointing out lipids are just not soluble in water. There's steroids, there's sphingolipids, there's triglycerides, there's phosphoglycerides. It's a gamish of stuff that isn't soluble in water. But saccharides have some order and structure, similar structure to them. Amino acids even more so. All right, you learn that, and you learn 6 of these R's, and you've learned the 6 amino acids that I'm talking about. They're not going to change right there. That's the general structure, and all we have to do is define that R. There are lots of amino acids, hundreds of them, but in the body there are 20 amino acids that are used to make these thousands of different proteins that any particular cell might need. They're building them off of 20 different amino acids. All right, so these are neat little molecules, and you can now relate from your organic chemistry where they're getting this name, amino acid. Okay, now you're going to see a little bit of difference right here. I got a COOH group over here. I have an NH2 group over here. At physiological pH of about 7.3, 7.4, this hydrogen dissociates off of the carboxyl group and it associates to the amine group. Okay? So when that happens, so depending on pH, pH of what the amino acid is floating around in will determine whether that hydrogen is sitting here or whether this hydrogen is sitting over here. Follow me? And if it leaves over here, it leaves as a proton. It leaves its electron behind. And so you get an O negative group over here. You get a COO negative group. And you get an NH3 plus group over here. So you got a +1 charge on the amine. You got a -1 charge on the carboxyl. Net 0. The molecule itself nets out to be neutral, but it has a positive and a negative end to it.
And that happens because of the pH?
Yeah, it happens because of the pH. So if you lower the pH, so it's 7.4, pH 7.4, actually, or you're greater than 7, really, that hydroxyl is coming off of there. Excuse me, the hydrogen is coming off of there and forming that carboxylate ion. As the pH goes down, as you lower the pH, you are increasing hydrogen ion concentration. So as you increase that hydrogen ion concentration, it will, there'll be more protons around. They will come on over there and get on that carboxyl group. And it won't be charged. So it is a function of pH.
You lower the pH, you increase hydrogen ion concentration, which attracts more.
Which provides more to go on that carboxyl group. And
That's when it would become an amino acid.
yeah, and then it would go back to that form. I got you right there. But in the bloodstream, now in the stomach is a different story. Stomach is low pH. But in the bloodstream and in the cell, inside the cell, it's going to probably exist like this. And we have a name for this. I think it's on the next slide. We refer to it as a zwitterion, the zwitterionic form of it. It's simultaneously negative and positive charged. One end of it is negative, the other end of it is positive. And that's the zwitterion. Not necessarily.
So is it to keep your body like neutral? Is that what that's for?
No.
How is it formed?
It just naturally formed as a function of pH, the hydrogen ion concentration. It just naturally does this. But it is helpful. You know, later on I'll talk about how these come together to form, when these come together to form a dipeptide, this is getting ahead of ourselves a little bit. But to form a dipeptide, one amino acid has to react with another amino acid. And they react like this. The carboxyl group reacts with the amine group. And you can see how this being negatively charged, that being positively charged, they're naturally being attracted to one another. In the way in which they need to bind to each other. This is how they need to react. This carboxyl needs to react with that amine group, and that is negatively charged, that's positively charged. These are clicking together. You know, these are like magnets just falling into place and then forming a dipeptide. We'll get into that later. But the charge on them is actually facilitating their combining to start building a protein molecule. Okay, so these are amino acids. We still haven't gotten to the R group. Okay, each amino acid has a characteristic side group or R group. The R groups are going to have different organic functional groups on them. It could be an aromatic ring, an OH. The R group could have another NH3+. In addition to the one that's on the amino acid, you could have an NH3+ down here somewhere. You can have another carboxyl group down there. All right, so I'm going to go, the next few slides are going to be keyed off of this list right here. We're going to classify all the amino acids into these 5 groups. And I mentioned that there's 6 amino acids I want you to learn. You're going to learn one from each one of these groups. And then one more that's just with the simplest one and is kind of unique in and of itself. So there's nonpolar. For the R group is neutral or nonpolar. Where the R group is polar, this would be an alcohol functional. You'd have an OH group on the R group. Aromatic. Positively charged R groups, where you have an NH2 or an NH3+. And negatively charged R groups, where you have a carboxyl that can lose that hydrogen and be actually a negatively charged R group. All right, so let's take these one at a time. So just think of 5 categories of amino acids. There's 20 amino acids the body uses. They fall into these 5 classes. So here's number 1, nonpolar R groups. Okay, glycine. So which ones do I want you to learn? This one. Glycine is the simplest amino acid. It just has, so R is an H. Okay, so just think about it. The R on glycine is another hydrogen. It's got a hydrogen there, got a hydrogen there. That's glycine. And then alanine, probably the next simplest one. All you got, the R group is a methyl group. That's the other one. So there are 2 in this category that I want you to learn. Glycine and alanine. And then these are the other ones. Proline, valine. See, valine has an isopropyl group on it. That's isopropyl. That's valine. Leucine, isoleucine. And you can see the way I drew that. I have the amino group on the left. I've got the carboxyl group on the right. And the hydrogen on the top and the R on the bottom. Remember, this can rotate around, so you can see it drawn this way. Amine, you got the carboxyl group. Those are both identical and legitimate ways of writing and drawing these molecules. Let's take a look at alanine a little bit more. We got a hydrogen. Here we have a methyl group. Is there a chiral carbon? There's only 2 carbons. Well, I guess there are 3 carbons. There are 3 carbons. The methyl, is there a chiral carbon on that molecule?
No. Yes. It's supposed to be.
Gabriel?
I would say yes.
What, that middle carbon? And why is it chiral? It has 4 things.
Oh, there we go. I was just counting the. There we go. Yeah.
And they're all different. Okay, so that's a chiral carbon. So this molecule is optically active. It's an optically active molecule.
That's alanine.
That's alanine. Okay, well, I point that out just to say that, okay, well, let's take a look at glycine. Take a look at glycine. Whitney, I'll give you another chance here. No. Because you look at this carbon right here, and you see two things that are the same. Okay, now, glycine is the only amino acid of these 20 that is not optically active. What I'm saying there is, so glycine does not have a mirror image isomer of it. Every other, the 19 other amino acids do. There is a, there's enantiomers. There's a pair of isomers that are mirror images of one another. I mean, yeah, we'll talk a little bit more about that in a minute. Let's look at all 20 of these. But to just see that this one has 1, 2, 3, 4 different things on it. This one, 1, 2, 3, 4 different things on it. All right? All of these, 1, 2, 3, 4 different things on that carbon. That is the optically active carbon. In each one of these, the alpha carbon is optically active in every case except glycine. All right, so glycine and alanine, and don't worry too much about it. I'll hand this out before you leave today. But I have, on the homework, I have you draw 6 amino acids. They are the 6 that I expect you to be able to draw. So you should make 3x5 cards. Yes? Well, means it has a chiral carbon. Because it has a chiral carbon, it will have a mirror image. And because it does, the one isomer will rotate polarized light one direction. The other one will rotate polarized. That's what we mean by optically active. It can be picked up on by polarized light in a polarimeter. So all of that stuff goes together. You got a chiral carbon, then you are going to have another isomer of that molecule that is a mirror image of that. And those two isomers, those two stereoisomers of one another are going to rotate polarized light in opposite directions. They're going to be optically active. Good question, Gabriel. I'm talking about it in, you know, all the pieces are out there. You just have to kind of attach them, right? That all goes together. If you have a chiral carbon, then you are going to have a mirror image isomer of that molecule, and those two isomers are going to be optically active. All right, so that's the first group. These things don't have any charges on them. They're nonpolar. These are nonpolar R groups. They're going to be hydrophobic. The R group itself is not going to be water-loving. Okay, polar. So now if you put an R group on there with a alcohol functional group, or this sort of a structure on it, there's going to be some polarity there, some water solubility. The OH bond is polar, and so is the SH bond. Look, at sulfur's right underneath oxygen. So it acts like an OH. That's a thiol. And this is the one over here. This is a critical amino acid in protein structure. Cysteine. And it's got the SH or thiol functional group on it. An SH group. Serine. We mentioned serine on Monday in lipids. when that can be an X group on phosphoglycerides. You can have serine on that phosphate. You have that amino acid I've mentioned. That's an amino acid we'll see. These are the polar. Aromatic. Phenylalanine. And this is where, we talked about PKU, phenylalanine. What's that stand for? Phenylalanine ketonuria, where you can't metabolize that particular, when an individual cannot metabolize that amino acid. Now look at, that's alanine. That is alanine. Come down here to alanine. But so if I had an H on here, that would be a methyl group, but we're going to put a phenyl group. And you have phenylalanine. Okay, don't make this too hard on yourself. Learn alanine, and then you say, okay, phenylalanine is alanine with a phenyl group on that methyl. It's phenylalanine. Tyrosine, tryptophan. So those are 3 aromatic. These are going to be hydrophobic. All right, they're basically non-polar, like alanine and the other in the other group, but these have aromatic rings. Positively charged. This is the 4th group. Positively charged. So you notice you've got this NH3 up here on the amino acid, but now you have 4 CH2 groups, and at the end of that you have another NH3 group. So the R group has the potential for picking up a positive charge. That's lysine. Yeah, you're going to want to know lysine. Like I said, these are all on here. We've gone through 5 already. Positively charged. And finally, negatively charged ones. The amino acid has a carboxyl group on it, but so does the R group. Have a carboxyl group on it. So we're talking about the negatively charged R group. Aspartate and glutamate is the one. Glutamate or glutamic acid. If I said it was glutamic acid, you'd have a COOH at the end, or glutamate where it has lost the H.
So glutamate is the one that we need to know?
Right. Well, that's your, I don't know, aspartame. I don't know, that molecule popped in there. Okay. You need to get a little bit familiar with the way we abbreviate these names. So alanine, there are 2 methods used. One is a 3-letter abbreviation, just basically shortening the name, alanine. So its shortened form of its name is ALA. Glycine, GLY. Phenylalanine, PHE.
The ones with the boxes are the ones that we're dealing with, or?
Well, we'll deal with all of those.
Okay.
We'll deal with all of those. Yeah, on your homework you'll have a problem here where you're piecing 4 of these together, and it's going to give you them with these abbreviations. It's not going to write out the whole name. I don't use this. There are textbooks that use it. It's just a letter. So alanine is A, arginine is R, phenylalanine is F. So, is that right? Which one is that? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7. Okay, yeah, it's F. So, phenylalanine. Giving it the letter F. So don't worry about this one. But if you're reading a paper and it's saying, okay, here is the amino acid sequence in this particular protein, and all it has is a series of letters, or it might have this sort of thing, you know, it might have dashes between there. It's giving you the amino acid sequence for the protein molecule. And to really, well, there's a lot you need to know to really elucidate a structure of a protein, but bare minimum, you need to know the order in which the amino acids are. It's like your address. You know, your address, my address has 5 numbers in it. And you can't just take those 5 numbers and scramble them, and I'm going to get to letter. The letters, the numbers have to be in the right order. Same way with the protein. You can't just have the right mix of amino acids. They have to be in exactly the right order, from the very first one to the very last one. Okay, we started talking about optical activity. I'm showing you glycine again right here. It is not optically active. It does not have a chiral carbon. But let's go over to something simple like alanine. Just have a methyl group. We have all of that, and the R group is a simple methyl. We've got a chiral carbon. Okay, now I didn't come here. I don't think I did. Maybe I did. Let's see. There's glyceraldehyde. Maybe I can even make an amino acid. Here's glyceraldehyde. And I mentioned this in chapter 16, that glyceraldehyde is the reference molecule for determining whether sugars are D or L. And all sugars, the naturally biologically active sugars, are D, right? Hasn't that? Remember me saying that? Say yes.
It says L on there.
What? Yeah. Well, the amino acids are always L. Sugars are D.
Oh, okay.
Okay? But glyceraldehyde is still our reference molecule, whether we're talking about sugars or amino acids. So let's see here. So here is L-glyceraldehyde. Here's L-glyceraldehyde. Remember, you put the carbons vertically. You put the aldehyde functional group at the top. You have the carbons fade away from you, and you say, okay, where is the OH and the H? The OH on the left, it's L-glyceraldehyde. Now let's see if I can make that. So I need to make, I'm going to tear apart my other glyceraldehyde here. Put a carboxyl group right here. I'm going to put a methyl group down here. Perfect. And then what do I need? I need an amine group right here. This won't take long. There's my amine functional group. Okay, Asenath, you want to help me?
Sure.
Come on up here. Come up here and help me.
So if the amine group is on the left hand of the protein, I just
Just
hold it.
hold your arm there. Stay out of the way. Just take that and hold that. That's glyceraldehyde. Okay, that Asenath has this molecule right here. I have alanine. This is alanine. I got a carboxyl group, a methyl group, my chiral carbon, and I'm going to arrange it the same way that is. The 3 carbons are going to be vertically, 3 carbons vertically. Then you ask the question, the amine group and the hydrogen. The amine group is off to the left. This is L-alanine. Okay? Because it mimics L-glyceraldehyde. All right, we're talking about a saccharide. Thank you, Asna. We're talking about a saccharide here, and we're talking about amino acid here. But we are arranging the carbons the same way, vertically. We're putting the carbon-oxygen double bond. This is a carboxyl group at the top. This is an aldehyde. We're having the chiral carbon things projecting towards us, and this amine group is on the same side as the OH is on glyceraldehyde. This is L-glyceraldehyde. This is L-alanine. And this is a biologically active amino acid, L. Okay? Don't get mixed up and think, well, sugars are D. Don't amino acids have to be D? No. No, this is the way we have set up a system to identify these things. This is the one nature uses, L-alanine. There is a D-alanine. That is the other isomer of this. But this is the isomer your body incorporates into protein, and this one, I don't know what it does with this one. It shouldn't see that one, because nature doesn't make this one. Shouldn't see it. And I put a shouldn't on there when there are synthesized forms of these molecules, you could have L and D. And this is either irrelevant, the body doesn't use it, or harmful, it gets in the way of things. All right, so that's all I want to mention here, but I do want you to know amino acids, all except glycine, have L and D isomers. And they all use the L. The L is the biologically active form of the isomer. So no matter what amino acid is, this methyl group, it's going to be some sort of an R down here. They're going to be arranged the same exact way. And the amine group on the left is L. If you go buy a bottle of lysine, you will find it labeled L-lysine. You should. Look at the ingredients, and it should be telling you it's L-lysine. Some people buy that amino acid as a supplement, and it is good when the manufacturer shows you that the form of it is all the L isomer form of it. Okay, those are just some more slides showing this optical nature. I can't believe it. We're already to the peptide bond. Okay. So, to make a dipeptide, yeah, Minnie?
Even the one who didn't understand, but how do you know it's an L or from the
Okay, how do you know? The distinguishing thing here, Minnie, is this amine group on the left. That is what distinguishes this as L. Okay, now Minnie, watch this. If I tear this off and switch this around, now I have to keep this arranged the same way. This amine group ends up on the right, and it's D-alanine. This is D-alanine. And this is what distinguishes them. This amine group being on the right when it is arranged this way means it's L. Okay? Does that help? Okay. Here, Pass that around. Everybody get your fingerprints on that. Okay, dipeptide. So if you take an amino acid and another amino acid, now here's the amine, the alpha carbon, the carboxyl group. The amine, the alpha carbon, the carboxyl group. If you look at that, you can split out a water molecule right here, and you're going to form a carbon-nitrogen bond. Carbon-nitrogen bond right here. And you have eliminated a water molecule from between the two amino acids. This, what functional group is that? From your organic chemistry. What is that functional group? What? An amide. It's not an amine. It's an amide. This is an amine, or an ammonium, but an amine functional group. This is an amide. You got a carbon-oxygen double bond right next to a nitrogen. We call it a peptide, or biochemists call it a peptide bond. Organic chemists would say it is a amide bond. All right, so I mean, we're in biochemistry right now, so we're going to just call it a peptide bond. And this is a dipeptide. It is a unit made up of two amino acids linked together right here. Now, this is important. I want you to appreciate this. We've got a nitrogen, an N+, on this end of this dipeptide. We have a carboxyl group on the other end. So we talk about it, an N-terminal or nitrogen terminal end to our peptide and a C-terminal end. Now let's just think about this for a second. If I came in here with another amino acid, let's put this as R3. If I came in here with another amino acid, we had a hydrogen here, and I split out another water molecule, I would still have a nitrogen on this end and a carbon on this end. Okay, what I want you to see, whether I've put 2 together, 3 together, 4 together, 5, 6, 7, 1,000, there's going to be a nitrogen on one end of the protein and a carbon on the other end. That's always the case. You get that? You see that?
No.
Okay, I did it once. We did it once here. We got this. We got a nitrogen here, and we got a carbon here. Okay? So you have to kind of look up here and see this. If we do it again, we take this unit. Well, let's take this unit down here, and we do it again down here. We're going to split out a water molecule, and we're going to this carbon is going to link up with this nitrogen to make another amide, and what's going to be left over? A carbon on the other end. So now we put 3 of them together, and that nitrogen isn't going anywhere. It's on the one end, and every time we put another one on here, there's going to be a carbon presenting itself on the other end.
So when it spits out that H2O, is that O at the end becoming like a double bond, and then it connects to
The O at one end. This one?
Well, that one gets shut off.
Yeah, it's water.
But
And
does that O turn into a double bonded O, and then it connects?
well, it is. This right here is this right there.
Oh, okay.
It is a double bonded O.
Okay.
It is a double bonded O. When we say COO, one of them is a carbon-oxygen double bond, and one is a carbon-oxygen single bond.
So, hold on, how does it become water? Does it
Well, this is a little. I mean, we've got the O8, we've got the hydrogen here, and we only have 2 there. Adelaida, I could see where you get a little confused right here because there's no hydrogen. But yeah, I don't want to try to get into that detail too much, but you could have this sort of thing. All right, you can have this sort of thing. You're getting 2 hydrogens off of the nitrogen and an oxygen off of the carboxyl. You still get water.
So where does water go?
So
The water just
is
floats
it a
around.
product? So water will always be a product?
Yeah, water will be a product of the synthesis of a.
You're taking the water out.
Yeah, it's dehydration. You're dehydrating between 2 amino acids and splitting out of water. Where's the water go? It just floats around in that medium. All this is happening in your cells of your body, which are in aqueous environment.
So we have to draw that in the test.
Your homework assignment asks you to draw a tetrapeptide. What's a tetrapeptide? It is a molecule like this with 4 of them. Tetra, 4. A quattro peptide, tetra, yeah, quattro. All right, so you get dipeptides, tripeptides, oligopeptides. You've heard this before. Oligomers, oligosaccharides. A very short 3 to 10 peptides would be an oligopeptide. A protein molecule is a polypeptide. On Monday, we're going to make a very small protein, insulin. And I think it has about 50 amino acids in it. That is a relatively small protein molecule. We're not going to make the whole molecule. We're going to make a portion of it. Unless you want to stay for 6 hours, we could try. We'd run out of kits. We couldn't make. We would run out of these molecular modeling kits to actually make the simplest, one of the simplest proteins, insulin. And we've got about 40 of these kits.
So if they're peptides or polypeptides with the smaller chains, and then the proteins and the ones that have more than 50 amino acids.
A protein is a polypeptide. It's poly,
With
many, more than 10.
More than 10?
Yeah, an oligo would be like 3 to 10 peptides. An oligopeptide. And they are important. There are some things like that floating around in the body. They're signaling molecules. They might actually be outside the cell, and they signal between the cells certain things. Okay, now here is a pentapeptide. So this is more complicated than what I'm asking you to do. One more amino acid complicated than what I'm asking you to do in the homework. But first of all, just zero in on this. This is the way you'd see it. This is the sequence. This is a pentapeptide with a serine, a glycine, a tyrosine, an alanine, and a valine. You can go back to that slide that shows these abbreviations if need be. This is very similar to the previous page. So right here is serine. From there over is serine. From this nitrogen to that carbon right there is glycine. From this nitrogen to that carbon right there is your tyrosine. You can see the tyrosine. It's not phenylalanine. It's like it. Got a phenyl group, but you also have an OH group on it. That's tyrosine. And then you've got alanine. And then finally over here you got valine. And you can see the R groups. You could draw, when you draw your tetrapeptide, you can draw the R groups above or below. You can see every alpha carbon has a hydrogen and something else. The glycine has a hydrogen, it has another hydrogen. That is a pentapeptide. Here is you see now we've got 5 in a row. We got a nitrogen on one end and a carboxyl on the other end. This is a little challenging. You have to pay attention to piece your tetrapeptide together properly. You're going nitrogen, alpha carbon, carboxyl. Nitrogen, alpha carbon, carboxyl. There is a peptide bond between each of the units. I will collect this homework at the end of class on Monday. You could probably do the back page at this point. You're going to find the front page deals a lot with the amino acid, the primary, secondary, tertiary structure, which is what will be the focus of our lecture Monday, or our activity Monday. So if you want to wait on that, it's up to you. That'll become very clear on Monday, the front page. There's some questions on the front page. It says name the 8 essential amino acids. You can go hunt those down. Asenath, I have some papers for you. What's the last date to withdraw? Friday.
This Friday.
What is today? The 14th. Friday.
Yeah, Friday.
Okay, if anybody has any questions about that, come and see me. But just be aware. What?
Do you drop or do we
You drop. Yeah, I don't drop. Don't count on me to drop. You have to do that.